So in this video, I'm going to look at GCSE biology again, focusing on AQA and specifically looking at photosynthesis, which is part of your bioenergetics topic area. So the chloroplast is the site of photosynthesis. Now you should know that this contains the chlorophyll, which is the green pigment which absorbs light energy. So you need to know the equation for photosynthesis, which is that water plus carbon dioxide are required by the plants so that they can photosynthesize to produce glucose and oxygen. Now, the water comes from the soil, the carbon dioxide comes from the atmosphere, and the sunlight energy is needed for this conversion to produce glucose and oxygen. So the chlorophyll is the green pigment that's going to absorb that light energy for the chemical reaction of photosynthesis to occur. So when you are drawing the chemical equation, you should always write the light energy above the arrow. Now you should know that this is an endothermic reaction, and the reason it's an endothermic reaction is because the energy is going to be transferred from the environment to the chloroplast by light. Now, you need to know that light actually will give you what's known as the inverse square law, that if I move a light source double the distance away from whatever it is that is doing that photosynthesizing, what's going to happen is the intensity of the light will be a quarter of the original. So how can I represent that? Well, if you look at my light source, which is my lamp, and I look at the distance, which is x, and we call that 1. If I move double the distance away, so 2x, what's going to happen is the light will spread out, and therefore the intensity now will be a quarter of the original. If I move further and double again, it's a quarter of the original. So if I go from 2x to 4x, the magnitude has changed. So it's important that if you're a higher level student, you know the inverse square law. If you move the light source double the distance, that intensity will be a quarter of the original distance. So why is that important? Why might they look at asking a questions about this? Well, it's part of your required practical activity number 5 to investigate the effect of light intensity on the rate of photosynthesis using an aquatic organism such as pondweed. So here I have my light source. I have my pondweed, which is in my boiling tube. I have a bung on top, and I have a probe which is going to either detect carbon dioxide levels within the solution, or it could be detecting the oxygen levels. That is then connected to a data logger. So the first thing you need to think about was, what is my independent variable? Well, in this question, they've told me that I'm investigating the effect of light intensity. So therefore, my independent variable is light intensity. Now, if I'm asked to describe this in the exam, I should always make sure that I give them some value. So I'm going to move the lamp different distances. I'm going to give them 5. So I'm going to move my lamp, and each time I move my lamp, I'm going to move it a specific distance, 0, 10, 20, 30, 40, 50 millimeters away from the pondweed. Therefore, I will be affecting the light intensity that the pondweed is being exposed to. Now remember, as I double it, so as I go from 10 millimeters to 20 millimeters, I have the inverse square law. I have quartered the intensity. As I go from 20 to 40, again, I have quartered the intensity. So by moving the lamp further away, I am affecting the light intensity that the plant is being exposed to. The other thing to note is about the dependent variable. Now, the dependent variable is the rate of photosynthesis. So how are you going to measure the rate of photosynthesis? And it's important that you note that the rate of photosynthesis is either going to be the gas collected over a given time, i.e. the gas collected would be oxygen, because that is a product of photosynthesis, or you could do it based on the rate of photosynthesis by the gas that's being used. How much is the carbon dioxide being used by the pondweed in a given time? So key thing to note that when it's rate, we always want to talk about that collection or how much of it's being used in a given time. The other thing you need to think about when it comes to required practicals are your control variables. So things like, what's the concentration of carbon dioxide? What was the volume of the water in the boiling tube to start with? What's the size of the pondweed? What's the size of the leaves? What's the temperature? Because these are factors that are going to affect the rate of photosynthesis. If we don't control them, that could mean that they are increasing or decreasing that rate of photosynthesis. Now, when you talk about using temperature, you always want to discuss using a water bath as a way in which you can control the temperature, keeping it at a level. Why is it important for temperature? Well, temperature is going to affect the kinetic energy of the reactants and any enzymes, and therefore if you don't control the temperature, that could mean that there's more kinetic energy, therefore more reactions occurring, or if there's too low a temperature, less reactions. So they do like to throw in a explain question on that one. So what does the plant do when it makes glucose? That glucose is a product of photosynthesis. What does it use the glucose for? Well, there are different ways in which the plant can use the glucose. It can use it in respiration. If you haven't seen my video on respiration, do check out how the complete oxidation of glucose will result in energy being released. So the plant could use the glucose in respiration. Now remember, respiration happens in all organisms. So whether we're talking about it being a plant, whether we're talking about it being an animal, respiration happens in all cells. Photosynthesis is very specific to plants and algae. So the glucose could be used in respiration, or the glucose might be used to make the cell wall, to strengthen the cell wall through the production of cellulose. It may be that it's stored in the polysaccharide, the large molecule of starch, which is an insoluble molecule and is a way in which the plant can store glucose for a later date. As we go into winter and the plants have lost their leaves, they still need to do respiration. So therefore they still need a supply of glucose. So the plant can store the glucose as starch so that it can use it when it's not able to photosynthesize as much. It can also use the glucose to produce fats and oils, another storage molecule. Or it could use the glucose to produce amino acids, and those amino acids are needed for protein synthesis. Now, it's important to note, to be able to make amino acids, the plant also needs to use nitrate ions that it absorbs from the soil. So they would be transported up through the xylem vessel with the water as it's being moved. So a few things to note then are limiting factors. So the factor that reduces the rate of photosynthesis when it is in short supply. So when you look at a graph and you are looking at, for example, the rate of photosynthesis against light intensity, up until the point in which the graph is not plateauing off, so up to the point before the plateau, whatever is at the bottom of the graph is the factor that is limiting the rate. So in this case, in each of my lines, you can see that the light intensity is the limiting factor for the rate of photosynthesis. Now, if I tell you that the first line represents low carbon dioxide concentration and a low temperature, and I tell you that the next line up says it's a high concentration of CO2 and low temperature, that means we can indicate that the limiting factor after the plateau in the first line was that the photosynthesis was being limited by the carbon dioxide concentration. After that point, the second line, what we're talking about is that the photosynthesis is now being limited by the temperature. And if we give it a high concentration of carbon dioxide and a high temperature, do not get caught out by saying that the enzymes would denature. High temperature does not necessarily mean we have reached the point of denaturation. So be careful with that. What you would want to say is that there is some other factor which is limiting that rate of photosynthesis, not the one where the enzymes have denatured. So students lose marks quite quickly by jumping to denaturation. So just be careful with that. So what would they do in the exam then? What they could do is they could give you a graph that looks like here. So here we can see the graph showing the effects of changing temperature, the light intensity, and carbon dioxide on the growth of the lettuce plant. And what they would want you to do is they would want you to be able to describe and explain what you can see. So the first one could be describe the effects of increasing light intensity at 0.03 carbon dioxide concentration and 25 degrees C. So it's very important that you take the time to look at that line and focus on what you can see. Now it's important to note that in this one we would say that there was a mean mass increase up to, give them the number, 3.5 arbitrary units. They've asked me to describe, so they want me to look at the graph and be specific. Therefore, up to that point, whatever was at the bottom of the graph, the light intensity was limiting the rate of photosynthesis. The other factors are limiting the rate after 3.5 as the mean mass levels off. So we can quite clearly say in that first one, to describe the effects of increasing light intensity, the effect of light intensity is that it's limiting it up to the point before the leveling off. After the leveling off, something else is limiting the rate of photosynthesis. Another question they could ask you to do is describe the effect of temperature on the rate of photosynthesis. So in this case, they want you to compare the difference between the 15 degrees C and the 25 degrees C. So by increasing the temperature from 15 degrees C to 25 degrees C, we've increased the mean mass of lettuce at each of the carbon dioxide concentrations. It doesn't matter if we're looking at 0.03% carbon dioxide or if we're looking at 4% carbon dioxide. Each time, my mean mass is higher on my graph. Therefore, we can say that a higher temperature increased the rate of photosynthesis, which resulted in a mean mass of the lettuce increasing. Now, when we think about that mass, what is it that mass could be? Well, that mass could be the starch that is being stored. It could be the lipids and the oils. It could be the proteins. So it's important that when you think about the mean mass, what are we talking about? We're talking about those glucose molecules being converted into starch, cellulose, lipids and oils, and proteins. If it is used in respiration, that wouldn't be adding to the mass. That would be where the conversion is going to be from that glucose being broken down to the water and carbon dioxide and releasing the energy.